So here is the second part of chapter 16, DNA replication. So here's Watson and Crick. They are the famous scientists that discovered that DNA is actually a double helix structure. So kind of a spiral staircase is the best way of describing it. So here's the quote again. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Pretty amazing discovery. So let's go ahead and look at the directionality of DNA, that it actually has a way that it has to go. So the way that we figure out the start and sort of the end is based upon the numbers of each carbon in the ribose ring here. So if you look over here, you have the 1 carbon, then the 2, then the 3 with a hydroxyl group, then the 4 bounded to another CH2 pairing, and then up here you have the phosphate molecule. So where you're going to see it's going to be 5' to 3'. So the DNA backbone, putting the DNA backbone together, refer to the 3' and the 5' ends of the DNA. The last trailing carbon down here at the end. So here's the 5' up top, and then the 3' will be down here. So here you have the phosphate. This oxygen is bound, so what's happening is the hydroxyl group that was here, you ended up with some water that was leaving, okay? So, again, this sounds trivial, but you'll see why this is so important in a second. So there's anti-parallel strands, meaning that they're parallel, but they're going in different directions. So that's what the anti means. Nucleotides in the DNA backbone are bonded from phosphate to sugar between the 3' and the 5' carbons. The DNA molecule has direction. So complementary strands run in the opposite directions. So here's 5' to 3'. Then it's the opposite on the other side. 3' at the top and then 5' at the bottom. So notice here how the letters are written upside down. So the direction is still 5' to 3'. You still would go this way. Just like this one is 5' to 3' in this direction with the letters written in this sequence. So bonding in DNA. Are they strong or weak bonds? And how do the bonds fit the mechanism for copying DNA? Well, let's look at the type of bonds that we have. Here you have hydrogen bonds, which we know are fairly weak bonds. And then here you have covalent phosphodiester bonds, which we know to be strong bonds. This means that these bonds here are going to break easily, and these bonds here are not going to break easily. So let's look at the base pairing in DNA. We have purines, adenine, which we abbreviate as A, guanine, which we abbreviate as G, and then the pyrimidines, we have thymine, which you abbreviate as T, and cytosine, which you abbreviate as C. Easy way to remember it is pyrimidines start with a Y, and thymine and cytosine each have a Y in it. Adenine and guanine are both would be purines without Ys. So the pairing, you're going to have A to T, adenine to thymine, and there's 2 hydrogen bonds, as you can see that here. So here's one, and then here's the other one to a nitrogen. So here we have cytosine and guanine is 3 hydrogen bonds. So you have 1 oxygen to hydrogen, hydrogen to nitrogen, and hydrogen to oxygen down here. So let's look at copying DNA, the replication of DNA. We know that the base pairing allows each strand to serve as the template for a new strand, meaning that it's semi-conserved. The new strand is half the parent template and half the new template. If you want to see how we discovered that it was semi-conservative, please see the 16 DNA history PowerPoint. So DNA replication. You have a large team of enzyme that coordinates the replication. So let's start here with number 1. Here we have the origin of replication and then this bubble that's created. Then the dark blue is the parental strand and the lighter sort of teal blue is the daughter strand. here we call this the replication fork. As we drop down, this is going to happen in multiple directions as it starts to get bigger until eventually you end up with 2 daughter DNA cells. So if we read this down here, in eukaryotes, DNA replication begins at many sites along the giant DNA molecule of each chromosome. In this micrograph, 3 replication bubbles are visible along the DNA of cultured Chinese hamster cells. The arrows indicate the direction of DNA replication at the 2 ends of each bubble. So here's one replication bubble, here's a second, and then here's a larger third one. So let's go ahead and see all of these different enzymes. So replication is the first step of replication. We have to unwind the DNA. We have to sort of open the zipper. In this case, it's going to be a helicase enzyme. So it unwinds part of the DNA helix. So here's the helicase. We actually have 2 of them. So there's one there, there's another one there. So we stabilize these openings by the single-stranded binding proteins, or SSB proteins. So you're going to have ones there, and here is our replication fork. So these 3 dots, or these 3 circles, is how we're going to represent the single-stranded binding proteins. So replication, here's the second step, that we have to build a daughter DNA strand. So we have to add a new complementary base to the one existing strand that there is. So we have to have an enzyme to do that. This is DNA polymerase 3. Okay, so here's DNA polymerase, and it's going to add the nucleotides. Okay, A to T. Okay, but we're missing something here. What could it possibly be? Of course, this, nothing's for free in biology, so where is the energy coming from? Well, let's look at this. The energy of replication. Where does energy for bonding usually come from? Well, down here we have ATP, and which is essentially just a modified nucleotide. Here's ADP. This is how we've known how ATP normally acts, is that you lose one phosphate. This becomes an inorganic phosphate, meaning that it's not bound to this carbon sugar anymore. And when that happens, we release energy. Okay, you remember ATP. Are there other ways to get energy out of it? So in other words, could we break a bond somewhere else to get that energy, or does it always have to be this end phosphate group? Well, you will see that in fact we can break it in the middle here. We're having 2 at the end, and so this will be A and P for adenosine monophosphate, and there is our nucleotide. Okay, so we can take the ATP molecule and modify it to create AMP. So we come with our own energy. Nucleotides essentially come with their own energy. So, and then they leave behind their nucleotide. So, does this mean that there are other energy nucleotides? Of course. So here's GDP, guanine triphosphate, is created into GMP, guanine monophosphate, and energy. So if you look at TTP, thymine triphosphate, and thymine monophosphate, and CTP, cytosine triphosphate, and cytosine monophosphate. So the nucleotides arrive as nucleosides. Nucleosides here are DNA bases with 3 phosphate groups, or the triphosphate. So PPP is the energy for bonding. We call them nucleosides. Make sure you're aware of that, and when you're reading through, to know the difference between a nucleoside and a nucleotide. Okay, so here's our nucleoside, 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 nucleoside. So the DNA bases arrive with their own energy source for bonding, and they're bounded again by DNA polymerase 3, which is an enzyme. So let's add these bases. You can only add the nucleotides to the 3' end of the growing DNA strand, which means that as the strand grows, it grows from the 5' to the 3'. So notice there's 2 different distinctions here. If it's the established strand, then you add it to the 3' end going to the 5th. As the subject of that statement, though, is the established strand. If the subject of your sentence is the strand that you're creating, then the created strand is going from the 5' to 3'. This is sometimes a source of confusion for students. So just realize, what is the subject? Is it the already established strand, or the parent strand, or is it the daughter strand that's being created? So let's move on. So we need a starter nucleotide to bond to, and the strand only grows. Again, this is the daughter strand we're talking about. Only grows in the 5' to 3'. So bring your own energy. Here we go. The energy rules for the process. So there is our nucleoside, DNA polymerase. It's going to cleave that phosphate bond there, and we just add our adenosine. Okay, so on with the guanine. Thymine to adenine. Cytosine to guanine. 5' to 3'. Again, notice over here the established or the parent strand is 3' to 5'. Okay, so let's say we're starting here at the very beginning. We have our strand that's together. We have to separate them, so this will become one parent strand, and this will become another parent strand. So again, the parent strand here is 3', then 5'. We're going to add it. This one will also be 5' over here, and 3'. Okay, so let's start to add again in the 5' to 3'. We're going to create a new strand, the daughter strand, in the 5' to 3'. So we need a primer base. So there's a guanine to cytosine. Okay, using the energy again as we go down. So it works out. Here we've just created it from 5' to 3'. So this is one of our strands. You're going to see this is called the leading strand in a second. So this one over here is 3' to 5'. So we have to start at the 5'. Oh, what are we doing? Is this going to work? Are we adding 3' to 5'? No, it's not going to work. We can't do this because you can't create this bond here. So you can't add 3' to 5'. So, because there's no energy to bond. So if you look, we're going to start here and work our way again from the 5' to the 3'. So, and then the next one will be cytosine bonding to guanine. Okay, and then we can start again all the way down at the bottom. Okay, again going from the 5' to the 3'. So you can see here's one big arrow, and we represent that as the leading strand over here on the right, and these shorter ones you're going to see are the lagging strand, and we call them Okazaki fragments. So let's learn more about this. Okay. Well, you might notice here that now this one is not connected, so we have another enzyme that we're going to add in here called ligase. Ligase is going to connect that bond, and then now that allows us to